a small recap on genetics, and then you're able to switch, and then you're able to do those other questions. So we say that genetics is it deal with genes. Whenever you talk about genetics, we are talking about genes. So in this, these are the topics which which we are supposed to cover in genetics. So I'm not going to go through each and everything. I'll go through them when I reach them. What is important here is to know the scientist who is the father of genetics. That is Gold Mendel, which was a priest. That one they can bring it in a objective and ask you a question. Which one of the following is the father of genetics? And then they give you Darwin. Darwin. They can say Lamarck. They can say Mendel. They can say James Watson. All those are scientists which we study about. But here in this case, we are looking for the person who was linked to genetics, who is Mendel. So they can ask you that question. And then, he said that he concluded that genes during his period, there were no genes. They were not called genes. They were called the factors. Factors, that these factors come in pairs and are inherited as distinct units. One from the father, one from the mother. The word gene just recently came to be known as genes, but previously he did not describe them as genes, he described them as factors. So if you are reading books and then you find factors, he was talking about genes. So what you need to know here, just a reminder, chromatin is coiled structure of DNA. If you look at a plate and you see spaghetti, that's how chromatin looks like in the nucleus. So we saw it like this. If this is the nucleus, and then that is chromatin network. Then you have chromosomes. Chromosomes are thread-like structures. Also, they carry genetic information. So a chromosome, everyone knows about a chromosome. Yeah, it's made up of two chromatids and then joined together by a centromere. So that's a chromosome. But when you pull it out, it forms what you call DNA. So chromosomes, DNA, chromatin network, all these things are almost the same thing on a different scale. You saying different areas, but in this case, those areas can be South Africa, Gauteng. Then you talk about Johannesburg. You could talk about Soweto or wherever you are. You see, you are just breaking down into smaller units. So chromatin network, chromosomes, genes, alleles, all these things are the same thing, but on a different scale. So when you talk about a gene, we say that is a portion of DNA that control a particular character. A portion or a part of DNA coding for a specific characteristic in an individual or a protein is what called a gene. So it means that DNA is long, but different sections, different segments that control a specific character is what called a gene. But a gene can all exist, can show itself in the phenotype in two different ways. For example, you have black and white. Black and white. Black is, yeah, it could be black, it could be an allele, and white could be an allele. But in this case, what is the gene? The gene becomes the color. The color of what? It can be a color of skin, color of eyes, color of what, depending on what are you looking at. So, if you are not black, you are white, meaning that a white is an allele, a black is an allele. What is that genogene? The genogene here is the color. So, another way in which a gene can express itself is what called allele. That's why we are saying that is each of two or more alternative forms, different forms of a gene found on the same locus, meaning that a gene can express itself in different ways. So we can bring that also in exam as a terminology. So these alleles, some are dominant, some are recessive. For example, the one which expresses itself in a phenotype in a heterozygous state is what you call a dominant allele. For example, if black is dominant over white, meaning that black is going to be capital B, while white is going to be, it won't be with a small w. No, because it's the same. So it's going to be small b. So the recessive one will take the small letter of the capital letter. I hope you understand, of a dominant allele. If the dominant allele is B, and then the small letter will indicate the recessive. So this is for the dominant allele, while this is for recessive allele. So in the presence of a dominant allele, recessive allele cannot express itself. So I think that one is also a little bit clear. We're just doing a highlight so that you're able to switch. Then you're saying that so recessive alleles can only express itself in a phenotype if it is in a homozygous state. So if it is capital letter, small letter, automatically the capital letter is going to mask the presence of the small letter, and then the individual is going to be black. Yes. So that's why they're saying that a heterozygous state, it can a dominant allele is that allele which can express itself in a heterozygous state, meaning that if it's like that, tall and short, maybe tall is capital letter and then short is small letter. So this individual is tall but is carrying a recessive allele, while the tall allele has masked the presence of the recessive allele. So that one is also clear. So we have conditions whereby if two alleles of the same kind exist, we call them homozygous. Homozygous. But if that allele is dominant, therefore it's called homozygous dominant. While the one, if it is recessive, then it's going to become homozygous recessive. If two alleles are different, therefore it's going to become heterozygous. Yes. So we are busy highlighting very fast. We remember that we say that phenotype is a description of an organism's in terms of physical appearance. Physical appearance, description of an organism in terms of physical appearance. So is what you call phenotype. If you look at someone, you say is big, is light skinned, he has a big head and big eyes. So you are describing someone what you see. Yes, the eyes are big, they are not hidden, we see them. The head is big, it's not hidden, we see it. The skin is light or dark skinned, it's not hidden, we see it. So what you see when you're describing someone, then it means that that is a phenotype. So when they ask you the phenotype of an individual, it means that what you see outside. Then the genotype, you are describing that type in terms of genes. Which gene type do you have? This genetic composition of an organism. So in this case, you look at into genes. You describe an individual in terms of genes. So if you look at this one, yes, which color is that? If it's blue or pink, I don't know. But inside we see that is capital B, capital B. So this is phenotype, while this is genotype. This is phenotype, this is genotype. Phenotype and then genotype. So when you describe someone in terms of, oh, they say the purple. You're describing someone in terms of what you see, then it's a phenotype. When you describe someone in terms of genetic composition, that is a genotype. Yes. We got the next terminology. Homozygous means alleles of the same kinds. If you have capital B, small b, it's the same kind. Therefore, is homozygous. While capital B, small b is meaning that they are different kinds, therefore it's heterozygous. What about capital B? Capital B, it's also homozygous. Therefore, because this is recessive, so it becomes homozygous recessive, or this one becomes homozygous dominant. Guys, remember that these are the words we use when it comes to the genetic cross. Then we go to the next one, which we call a monohybrid cross. Monohybrid cross means you are crossing an individual, but using one characteristic. Yes, for example, height. If you are looking at someone's height, yes, or what you call height, whatever, I don't care. So in this case, you look at the person is either short or is tall. Yes. If it's tall, cool. If it's short, cool. So one characteristic is going to be considered here. That is height. So the person can be tall or short. That is alleles, either tall or short. So if I consider this in our genetic cross, therefore it's going to become mono. So the result when you cross is what called a hybrid. That's why they call it mono hybrid. So it's a cross which involve one characteristic. So Mendel's law of segregation, you need to know it. This one is talking about separation. When do they separate? They separate during meiosis. He says that that diploid organisms like us, we are 2N. Remember the 2N, it depends on the organism, how many chromosomes do they have. It can be 10, it can be 20, it can be 46, like human beings. Yes. So it depends on how many chromosomes does individual have. So these diploid organisms pass randomly selected alleles. You don't select at which allele. You did not choose to resemble the way you are resembling. If you resemble your father, you did not choose it. Even your father did not choose it. So they just do it randomly. You don't know which sperm is going to fertilize the ovum. You don't know which allele is in there. So they happen randomly for that specific what? Trait. So each individual will have two alleles. Each allele, one will come from the father and one come from the mother, meaning that each parent must contribute one allele. If you are tall, one allele came from the father, one allele came from the mother. So that's what he's trying to explain in this regard. And this separation, it happens in such a way that you don't have any influence about it. It happens randomly. The next point which we talked about is the format for representing a genetic cross. We said that this is how we mark. This is how we mark our genetic cross. So if you're marking our genetic cross, remember, you start with the P1, F1. Guys, I say that the moment you like P1, F1, we shall do this and we give you a mark. Whether you wrote this or not, or they are correct or wrong, as long as you wrote P1, F1, we'll give you a mark. So I cannot accept someone lose that mark because that mark is mahala. Number two, you write phenotype, genotype, meiosis, and then you also write the word fertilization. You writing meiosis and fertilization, we give you another mark. We don't care whether this is correct or wrong. We give you a mark. You have written meiosis and fertilization. So those are 2 marks out of 6. So the third one, remember we say that the phenotype. Phenotype, we get it from the question. They will tell you that Zintle, who is light-skinned, married Karabo, who is dark-skinned. You see, they said it. They said it that a person who is light-skinned married a person who is dark-skinned. You see, so that is another mark. You just get it from the question, put it here, you get another mark. So you have 3 out of 6, meaning that on any genetic cross they bring, you have 3 marks mahala. There is no way you can tell me that you get 0 when you have such. We continue. So from there, the moment now you know this, you have got this, you have got the one for meiosis. Now the genotype, they'll specify either is heterozygous or is homozygous or is recessive, whatever. They will tell you which one is dominant, which one is recessive. This is where the problem comes from. If you get it right, you are most likely to get the whole question right. If you get it wrong, you are most likely to get the whole question wrong. So what do is you have to make sure that you get these answers right, most especially this point, so that you're able to get 6 marks. However, when the question is asking you the ratio or the percentage or yes, make sure that you get that correct, that answer correct, because it might be a compulsory mark. which we mark and we put a C or we put a star on it, which means that is a compulsory mark. If you get all of them, you get 7, but you did not get the compulsory mark, you will get 5. We will first count 1, 2, 3, 4, 5, and then we look for the compulsory mark. If the compulsory mark is on there, even if you have got 7, we shall give you 5. Why? Because you got the compulsory mark wrong. So the compulsory mark, in most cases, we see it when we ask you a question. Identify the phenotypes of the offsprings. The phenotypes of the offsprings are most likely to be the compulsory mark. So you have to make sure that you get it right. I think I'm done there. So that's how we mark. We P1, F1, we give you a mark. Meiosis and fertilization, we give you a mark. You write a phenotype, we give you a mark. You write a genotype. And I said this is where the problems is. You get another mark. Then when you cross them, either you are using a Punnett square method or you're just using crosses, still we give you another mark. After that, you write the genotypes and the phenotypes, we give you another mark. So guys, as long as you interpret the question properly, you will be able to get answers regarding to genetics. Genetics is not that hard as long as you know how to interpret that question. Codominance. Let's talk about the codominance until we reach our blood group where we, yes, dihybrid, yes. When you say dominancy, it means that you are being super. You are like you have more powers over others. So if you are dominant, you can have complete power. Yes, father and mother. Who is the powerful in the house? Is the father. If you are the mother and you are the powerful, check yourself. We are talking about science. All right. We are saying that father and mother, okay, students and teachers, who is the most powerful in the class, automatically is the teacher. President and vice president, who is the most powerful? You will find out that it's the president. If the president is there, the vice president cannot talk. For the vice president will be regarded as a recessive, while the president is going to be regarded as dominant. So if the power you have makes another one to be silenced, which we call masked, yes, we shall say that that kind of cross involves is complete dominance. That's why if you find a genetic cross involving a capital letter and a small letter, then that is complete dominance. Then you have the incomplete dominance. You are dominant, you have the powers, but you don't have powers over other people. You understand? You have powers. Your father in your house has no power over the neighbor's house. No, you or his neighbor. No. However much he has powers and the neighbor has power in his house, but both of them, if they sit on the same table, they'll have equal powers. Meaning that if they are discussing about something, this one must bring 50 and the other one must bring 50, so that you form something in the middle. It's not the other one, it's not the other one. Meaning that if you have black, you cross it with white. When you mix the two, you don't produce black, you don't produce white, but you produce an intermediate, which is gray. You understand? So here they're saying none of the two alleles of a gene are dominant over the other, meaning that they have the same powers, resulting in an intermediate. Black, white, you produce gray. Please don't forget about it. And what you need to remember here is complete dominance has capital letters. They use capital letters. So if it is black, it's going to be capital B, and white is going to be capital W. But here, check. If it is tall or black is dominant over white, black is going to be capital B, white is going to be small b. You see why? Because black is completely dominant over white. While here, black is dominant and white is also dominant. Therefore, they produce 50-50. And when I combine the 50-50, I produce a gray. Not black, not white. Yes. However, black can also be produced and white can also be produced, but an intermediate must also be produced. Lastly, is co-dominance. When we say the word co means together, meaning that you are together. For example, if your father has a wife, which you call the first lady, yes, but your father might have another, which you call the roll on, the side, whatever you call it, I don't know. Meaning that the other one is a second wife. So these two wives, they coexist. You understand? So when you say co, it means that together you exist at the same time. So if your dominance is co, it means that your power exists at the same time. You show what you are, even the other one shows what he or is. You understand? So it is a genetic cross involving both alleles are being equally expressed in the phenotype. So if it's black and white, you produce an individual with black and white at the same time. You have black, you have white, you have black, white patches, and then black patches. You see? So it means that both alleles are dominant. Still, you will see that this is black capital B and W is capital W. So it means that complete dominant. In complete dominance and codominance, when you look at the way how you cross, they have the same letters. But the only difference which is there is this one produces an intermediate, while this one produces a mixture of the two alleles or the two pillars. Let me, before I continue, Ariel, are we together? Ariel is not online. Chico, are we together?
Yes, we're together.
Okay. Uh,
Yes, sir.
Kensani, are we together? Zintle, are we together? Zintle, where are you?
Yes,
Kensani,
sir.
where?
Yeah.
Nkensani, where are you? Hey, don't tell me that I'm like a radio. Like, you just switched on there and then you relax yourself somewhere else. Ah, where's Georgia? Georgia, I don't see Georgia. Mm. So, we continue, guys. We continue. Okay. So this is what I was saying. You have red crossed with white. Is it white? I think it's white. I don't know. You produce a red, meaning that the red is dominant over white. You see that you have small letter, you have capital letter. Yes. And then uh red and blue, you produce red and blue together at the same time, meaning that that is co-dominance. They exist at the same time. While red and white, you produce pink. You don't produce red, you don't produce white, but you produce pink, an intermediate. So it means that that is incomplete dominancy. So you have to know that. We continue. So this is an example of a genetic cross. They crossed allele for tallness is capital T, is dominant over allele. You see, they specified it. Tall. So you see now we're gonna find out. A man who is homozygous. You see this word homozygous? Yeah, tall. So I've got the phenotype. Marries a woman who is homozygous short. You see? Meaning that now I've got the tall and the short. But they say this is homozygous, meaning it's going to be capital T, capital T. Obviously, if you are short, automatically, in this case, because tall is dominant over short, it's going to be small t, because the moment you have say capital T, small t, it's going to be tall. So even if I don't say homozygous, so I will know that, oh, short is small t, small t. After that, meiosis must take place. We circle this. If you don't circle them, yes, meiosis must take place. When meiosis takes place, what is next? You form the gametes. The gametes, this crosses with that, this crosses with that, this crosses with that, this crosses that. Make sure that these things are linked. Don't just cross and then leave them in space. Or use this. Then we shall come, we say here, we give you a mark, we give you a mark for fertilization, we give you a mark there. That is 3 out of 6. Already you have them there. Then you have identified this properly. Then we give you another mark, we give you another mark, and we give you another mark. So we will look only for 6. If worst goes to worst, it will be 7, but it's very rare. These days, we're no longer giving it 7 marks because we see most of the people will get that marks. So we don't need to repeat giving you a lot of marks for a simple thing. Okay, we continue. So is the complete dominancy, you see that we say the complete dominancy, incomplete dominancy, both are capital letters, you see, both are capital letters, but because it is incomplete, so I will need to produce an intermediate. Say the red crossed with white, what did they produce? They produced pink, which is an intermediate. That's what will help you to know whether is complete dominancy, sorry, is incomplete dominancy or codominance. Look at this codominance. You see it's red and white. What did you produce? A rod, which is a mixture of red and white. But here, what did we produce? We produced pink, which is an intermediate color. So that's the difference between incomplete and co-dominance. Yes. This one is what I've been explaining. Then this one is about sex inheritance. We described this yesterday when you are trying to explain how is the father determining the sperm or the male gamete determine the gender of an individual. So basically, a man who marries a woman and then have children using the genetic cross determine the possible genotypes and phenotypes of the children. Meaning that now, if they say possible genotypes, this could be a compulsory mark and this could be a compulsory mark. If you don't get them right, even if you have got the rest, we will first subtract that. If it is out of 6, you'll get 4. So P1, a mark, a man, check. A man marries a woman. You see, this is always obtained from the question. But a man is XX, XY, while a lady is XY, XX. So meiosis takes place. After meiosis taking place, you have that. So they are being separated by a comma. What next? Fertilization must take place. So this goes with this. That's why you have this. This with this. That's why you have this. This with this. That's why you have this. This with this. That's why you have this. So now you're going to ask me, why don't I start with Y? I think we refer Y as a recessive. We refer Y as a recessive. So meaning that this is a female, male, male. I have 2 males, 2 females, 2 males. So basically, if this, the ratio is going to be 1 to 1. Don't forget, guys, that I told you that once you get a ratio of 2 to 2, reduce it to 1 to 1. If all of them are boys, for example, what's the ratio? It's 1. Not 1 to 1. 1 to 1 is 50. We got the next one. Yeah, if they ask you, they can bring 6 marks and they ask you to describe how sex is determined in humans. 20 pairs of chromosomes in humans are autosomes. 1 pair is called a gonosome, or what you call a sex chromosome. Males have X and Y, while females have X and X. If a sperm carrying a Y chromosome fertilizes the ovum carrying the X chromosome, then the individual is going to be XY. The individual is going to be a male. If a sperm carrying X chromosome fertilizes the ovum with X chromosome, then the individual is going to be XX, meaning that ladies are always bringing X, so they cannot determine the gender. It's only the male who brings either X or Y, depending on what they have brought. That will determine the what? The gender. That's why is if your mother is producing only girls, your father must not go and get married to another lady claiming that you are giving only girls. The problem is you, the guy, because it's the guy who determines the gender of an individual. However, how sure are you that the female might kill all the Y chromosomes, sperms with Y chromosomes? So it could also be an issue. Anyway, if you, sorry, yes, Zintle? Zintle?
No, it's not me, so I didn't say anything. I think someone.
Hello, sir.
Kiko, yes.
My question is, right, why is it in most cases, right, when men, right, let's say if a woman who's only giving birth to children who are girls, right, when they change, they'll have a boy. Why is it like that? In most cases.
That's why I say that, hey, it could be the problem of the lady, yes, whereby maybe the sperms with Y chromosome are what? Are being killed. However, it might not be. Remember, producing a boy, a girl, is like betting. It's like betting. You have 50% chance of having a boy or 50% chance of having a girl. So it's like you are betting. I'm not good in betting because I don't bet, but they might be kicking you whenever you are betting with this betting. How do they call it? I don't know. This betting ish. I don't know how they call it. Ne? Where they bet from. You're betting from here, they're kicking you, they're killing you. Whenever you put there, nothing is happening. When you change the betting ground, then you go to another, maybe betting what. You start winning. Oban. So it could be, remember, this thing is chance. You have over 500 million sperms. You don't know which one is going to fertilize the ovum. But maybe 250 million sperms are male, 250 million sperms are female. And then now, which one is going to fertilize the ovum? We don't know. So this thing of boys, girls, or boys, or girls, there is no formula for that. If you change, you change. You're just by luck that you produce a different gender. But you saying that, no, this lady is only giving me girls. Hey, the problem might be you, the guy. Why? Because you determine the gender. You either produce X or you produce Y to this individual. And then now that one is just receiving and adds on what is already having. And then automatically the gender of an individual is determined. So when they change, it could be I don't know. I don't know. It could be a good luck. Uh-huh. So we have what you call sex-linked inheritance. Also saw this sex-linked inheritance. Sex-linked inheritance, these are inheritance or these are when you talk about sex-linked genes, these are genes which are carried on X chromosome or sex chromosome. The reason why we say X chromosome because Y doesn't carry any gene. So we probably say that you carry on X chromosome or sex chromosome. Um, these genes which are carried on this sex chromosome or X chromosome, what they determine is what called the sex-linked characteristic. So, for example, you have this X, you have a gene, ne? A gene is carried on this X chromosome. So, what this gene is determining, we shall say that it's sex-linked trait. sex-linked trait or sex-linked character. So characteristics whose genes are carried on X chromosome or sex chromosome are defined as sex-linked characteristics. And those genes which are carried on X chromosome are called sex-linked genes or sex-linked alleles. Examples is hemophilia, that is the failure of the blood to clot. Inability of the blood to clot is what you call hemophilia. And then color blindness is the failure of an individual to differentiate between the primary colors. Yes, we could say that you are color blind. Night blindness is also an example, but we don't usually ask you about it. That's why I underlined when I was writing this book, I underlined this and this, which means hemophilia and the color blindness are the mostly to be asked. But guys, there is something which you need to note. A question will always come and they will say, John married Josephine or married, let me say, Sarah, because I've never seen Sarah who is a male. So if John marries Sarah, and they did not say that what they have is sex linked, please don't use X and Y. Please. It means that that character is not sex linked. We shall see those examples. So we must always look for the word is sex linked or is carried on X chromosome or is carried on X on sex chromosome. So it must have that word which is linked to sex linked. If they don't talk about it, then it means that they're going to ask you a question if it's sex linked or is non-sex linked. And then now you have to find out from whatever statement they have given you, there will be a clue to show you that, oh, is sex linked or is non-sex linked. The clue will be there. And that clue, use that clue so that you are able to identify whether it's sex linked or non-sex linked. If not, please don't use X and Y. However much they have talked about John and they have talked about Sarah, and you know that John is a male and Sarah is a female. Don't use XX here, don't use XY because it is not sexy linked. Let's have an example of a sex-linked trait. Hemophilia is caused by a recessive allele on X. Are you seeing? They have specified it. That hemophilia is caused by a recessive allele. Is it sex-linked? What shows? On X chromosome. You see? Small h, a man who has hemophilia marries a lady who is normal but a carrier and produce children using the genetic cross, show the phenotype and the genotype of the children. First step before you think P1, F1, so that you're able to get that mark. Second step, you write phenotype, genotype, meiosis, fertilized gametes. Yes. Fertilization. Then so that you're able to give you another mark. Third step, you go back to the question. They said, who married who? A man who is hemophilic married a lady who is normal. So I will say male who is hemophilic married a normal female. Do I need to say carrier? Carrier is just a statement which is going to help us to know if he's homozygous or is heterozygous. If he's a carrier, it means that is heterozygous. So now I think we got that mark. Now we have 3 marks. Now we come here. We know that a male is XY, but Y doesn't carry anything. So, but they say the man is hemophilic. And the hemophilia is caused by a recessive allele. That's why I put is small h and then a capital Y. Are you seeing? Well, a lady is XX, yes, but they said is normal. So I expect to have a capital H. If they just say normal, it means that even this one is going to be capital H. But they said normal, but a carrier, meaning that is carrying a recessive allele. Therefore, I'll come here and I'll add a recessive allele. Are you seeing? So it means that I have this and I have this. If I have this and I have this, that's what we have here. Then now meiosis is going to take place. I form the gamete, I form a gamete, I form a gamete, I form a gamete. After that, what next? I will either use a cross or I will put this in the Punnett square method box. So bring this, I put it here, bring this, I put it there. Bring this, I put it there, bring this, I put it there. After that, I do my genetic cross. So this goes with this, capital H, small h, you see. This goes with this, capital H and a Y, you see. This goes with this, small h, small h. This goes with this, small h and Y. So meaning that I have two females and two males. However, one female is hemophilic, is not sick. Sick of what? Sick of what? You don't just say sick. If you say sick, is affected. Affected with what? The question is saying hemophilic. These people are suffering from hemophilia. Therefore, this individual is a female who has hemophilia. That's why you're saying that hemophilic female. That's one. Then this one is normal, but a carrier. But I don't need to specify that he's a carrier. Is normal. Carrier is only helping us to know whether it's heterozygous or homozygous. Therefore, I will say a normal female. Then we have this guy who is hemophilic. Hemophilic. This is supposed to be hemophilic male. And then I have the last one, which is the normal male. Yes. So that's how you're supposed to do that. So I'm going to give you,
So
yes,
the previous questions, how will you indicate the ratios?
the ratio. The ratio in terms of what? Do you mean phenotypic ratio or genotypic ratio?
Like as it is, isn't it? That
You see, all of
example.
them are different. So it means that it's going to be 1 to 1 to 1 to 1. Yes. Unless they are looking for the ratio for sick individuals. You see, if they say sick individuals or hemophilic individuals, they're not saying hemophilic male or hemophilic female. Babon. If
Yes.
they are just generalizing normal versus the hemophilic, if it's normal, this is normal, and then this is normal. So I have two normal and two hemophilic. So the ratio is going to become 1 to 1. But if they're just saying the phenotypic ratio, then automatically all of them are different. This is normal, but female. This is hemophilic. This is a female, and then this is normal, but it's a male, and then this is hemophilic is a male. So the ratio is gonna be 1 to 1 to 1 to 1.
Okay. So, but then if they don't ask for the ratio, it's not compulsory, right, to write the ratio to indicate it.
I prefer to write it because sometimes it could be a marking point, but you have lost somewhere other marks, and it can help you to top up. So yeah, it's better to write everything. You know, when it's not we are not marking the first correct answer, then you can write whatever you have in the head. Don't limit yourself because you don't know exactly what they're gonna look for in that question. So it's better to write everything and then You leave it there for the examiner to mark. The examiner will mark. If the marks reach 6, for example, the question is out of 6 and they are looking for 6, they will stop marking. But if you leave it, you still have time, and then you end up sleeping in exam, and then what you wrote is 4, then it means that you're going to lose a mark. Yet you'd have got 2 marks extra if you had written it. So I prefer to write everything. If the question is not limiting you, that list 2 or give 2 or give tabulate 2 differences. As long as they are not limited to that specific number, write everything. But if it is limited to that specific number, then it means that you have to write what is the most correct, what you think is most correct first. And then the one which you are not so sure, you write them last. Because we're going to mark the first one.
Okay. Okay.
Yes. So, I will send this to you guys. You're gonna practice it for me. You'll send it to me. So I'm sending it in the group, and those who are not in the group, I'll send it to individual people so that you're able to do it for me, and then I see the way forward. Yeah. Okay. We go to blood group. We go to bloody group, blood group, blood group. All right, I've sent it to you to you will do it for me. All right. Blood group, we also talked about it yesterday. Remember? Here, when you talk about blood grouping, you have to look at how many genes, how many alleles, how many genotypes, and how many phenotypes. If you know, then if I have blood group A and blood group B, can I get blood group O? If I have blood group A and another one is blood group AB, can they produce a kid of blood group O? So you just have to know, if I mix this and this, what do I get? If I mix this and this, what do I get? So if the question can come when it's not asking you to mix, but indirectly is asking you to mix, meaning that to find out if it is possible. Like that question we did yesterday, which was saying that the father, I think the mother was blood group B, and then the father is blood group A, B, and then the child is blood group O. And then we said that now that one is just helping us to kick out some individuals who can't be the father of that child who has that blood group. So blood grouping is an example of a character with multiple alleles. There are 3 different alternative forms of genes for blood group, meaning that 3 alleles controlling a blood group. And all these alleles, they occupy the same locus. A locus is the position of a gene along a chromosome. That's what we call a locus. If there are many, we call them loci. Position of a gene on a chromosome. However, only 2 alleles can exist in an individual because you get one from the father, one from the mother. So we see that blood group A has antigens A. That's why you're using I, antigen A, antigen A. And then it has a recessive antigen, meaning that it has no antigen there. So you can be homozygous or heterozygous. Just a moment. You can be homozygous or you can be heterozygous. So if you're homozygous, then it means that you're going to be like this. If you're heterozygous, you're going to be like that. So blood group A has, look at the antigens they have. Blood group B, check the color. Blood group AB has the mixture of A and B. Blood group O has nothing. You know, I always laugh at people who say that blood group O is the strongest. Who told you? Who told you that blood group O is the strongest? Strongest where? In your family. Blood group O has no antigens. That's why they call it blood group 0. You have nothing related to antigens. So because you don't have anything on your structure, cell structure, you can pour that blood group to anyone. Therefore, even O itself. So blood group O is regarded as a universal donor. It can donate anyone without agglutination, without clotting of blood. That's the meaning. But blood group AB has for A and has for B, meaning that it can accept A, it can accept B, it can accept itself, and it can also accept O because O has nothing related to antigen. Therefore, blood group AB becomes the universal recipient. It can receive from anybody. While blood group O is universal donor. It can donate to anybody. If you can donate to anybody, but you can't receive from anybody, then you are worse. You are worse because if you don't have blood group, you don't have blood, nobody is around you having blood group O, then you suffer the consequences. But if you're blood group AB, anybody around you can give you the blood. And then your life is saved. So blood group AB, I think, is the best. Anyway, doesn't matter. So, how is the person of blood group A marries a person of blood group A and produces a kid of blood group O? That can only happen if blood group A is heterozygous. The mother is blood group A heterozygous and the father is blood group A heterozygous. So this recessive can combine to produce blood group O. Imagine before, people couldn't know this science behind. And there are so many people who were accused of fornicating or committing adultery in the families because the father is blood group A, the mother is blood group B, and the child is blood group O. They say that is impossible. It can't be. You have to find out who is the father of that child. But nowadays, it's possible because you can be heterozygous or you can be homozygous. So anyway, it's possible. Okay. All right. We continue. Yeah. We said that there are three alleles, allele for A, allele for B, and the allele for O. And then you have four types of blood groups, A, B, AB, O. So we call this homozygous, heterozygous, homozygous, heterozygous. How about this? And this is homozygous. So when they say blood group A heterozygous, I think now you know what it is. When they say blood group B heterozygous, you know what it is. So actually, if you go back to dominancy, this is complete dominance because it has a capital letter and a small letter. This is also complete dominance. What about this? This is co-dominance because A and B, they exist at the same time. When you are marking, you say I and you do like this, we'll give you a 0. When you're marking, you say I like this, we'll give you a 0. The I must be the I, not a Roman figure, which means 1. Please make sure that you don't lose those marks. So here is the question. Susan with blood group AB marries John with blood group O. What will be the possible genotype of their offspring? So you have to do Susan blood group A, and then that O, AB, and O. We have the phenotypes. So you have this mark, you have this mark. So those are the 3 marks we talked about. So we're only looking for 3 marks. So you guys, you cannot fail to get that. Let me take this question and also send it to you. Yes, I'm sending you the second question. You're gonna practice for me. You're gonna practice for me. Okay, I'm sending you another question. Another question. You're gonna practice for me. Yeah, I've sent it. Yes. Dihybrid cross. Yes. Any question, guys? Zinhle.
No, sir.
Kensani. Kensani, where are you? Kensani. Last time I asked you, you didn't talk. Maybe you were there far from your phone. So do you have any question?
No, sir.
Oh. Erio?
No, sir.
Erio, why are you having too much energy? All right. Zinhle?
No question.
No question. So you
Yeah.
guys, if I set a paper on what we have discussed now, can you give me marks?
100%, sir.
100%. Okay. I'm going to we discuss this, ne? So today we are supposed, yes, we discussed this, we discussed, I think, okay. Yeah, we're supposed to start from mutations. All right. Let me use a few minutes to get few here. Then after that, we're going to have to practice a lot of questions concerning about this. But now that is not going to stop us from going to the next topic. At least we're going to have to shift to the next topic. But as we are shifting to the next topic, we do one question from the previous, then we continue to the next topic like that. Because if you don't practice genetics, things might be worse on your side. Yeah, then when you talk about dihybrid, this is the genetic cross which involves one character. Yes, genetic cross involving one character. Here I will give you more questions about dihybrid because the way how we teach it is not the same way how we set it. A dihybrid cross involves inheritance of 2 characteristics. So we say that monohybrid involves 1 character. Dihybrid involves 2 because di means 2. So if you have color and you have size, so you combine color black and then white and then size big and then small, meaning that I have 4 alleles. So when I'm forming the gametes, each individual must form 4, meaning that I'm going to have to be with 8 gametes. But when these gametes are forming, when these gametes are separating, meiosis is taking place, and these gametes, they do so independently. No gamete is going to influence the other. For example, your size is not influenced by the size of your body is not influenced by the size of your head. The size of your body is not influenced or is not influencing the skin color. The size of your body is not linked to the intelligence you have. Why? Because those are two different characters. And when meiosis is taking place, they segregate, they separate, they segregate independently. So one character does not influence the presence of another character. So Mendel explained this using the principle of independent assortment when he was describing the garden peas. What did he do? We say that he crossed yellow and round with the green and wrinkled. When he crossed them, he found out that all of them are round and yellow. Then the question can come and ask you, which one is dominant and which one is recessive? You crossed yellow and round with wrinkled and green. Then after that, you produce yellow and round. It means yellow is dominant over green and round is dominant over wrinkled. You see? So they just say that something like that and they ask you a question. What is what? You see the first question where it's coming from? That's number one. Number two, they can give you that they crossed yellow and yellow and round crossed with black and wrinkled. Here they used black. It's fine. They ask you, write down the possible gametes from each individual or each parent. So I will come here. Sorry. Yeah, I will come here. You said you crossed what and what? You crossed yellow, of which yellow is capital Y, yellow and round, capital R. And then the other one is black and wrinkled. So they're asking you to write down the gametes. So it means that the gametes are supposed to be 4. If it is monohybrid, it forms 2. If it's dihybrid, it forms 4. So this is going to go with this. So I'm going to have capital Y. Goes with this, capital R. That's the first gamete. Then this with this, capital Y, capital R. So now this one is done. It has gone with the 2. Then now this with this, capital Y, capital R. And this with this, capital Y, capital R. So I formed 4 of them. What about this? It's going to be capital small y. They are supposed to be aligned, you know? So I don't want stories. It's going to be this with this, small y, small r. Then this with this, small y, small r. Then this with this, small y, small r. Then this with this, small y, small r. They are formed also. So because all of them, so this is because all of them, they are the same. So it's going to be capital R, capital Y, capital R, and then capital small r, Y, small r, because all of them, they are same. That's why you'll find out that they'll only give you 2 marks for finding out all this. But because all of them are the same, just take 1. So it means that when you combine the 2, it's going to be, remember, you're supposed to combine this and this. You do a Punnett square, but all of them, because they're the same, meaning that I'm going to have to get the same result. Capital Y, small y, capital R, small r. This with this with this. Simple. That's why here in our booklet, we abbreviated it. We abbreviated it that when you cross this and this, you produce this. It's the same thing here. Meaning that all of them are going to be like that. So now this is the F1, P1, F1. But now this is P2. They brought this one, the result, then they cross them again. So it's going to be P, P2, F2. Second parent. So if I cross, they still can ask you identify the gametes in this. So it means that this is going to go with this. So I'm going to form capital Y, capital R, that is the first gamete. Now this with this, capital Y, small r. Now this with this, small y, small r to r. Why am I saying with the small y? Because this y is a different character from the r. So the y is not influencing what is r is doing, and the r doesn't influence the what is y is doing. That's why now this time, if it was R, capital small r, then I would have started with the capital R. But now Y and R are totally different. So now this one with this small r, small y, small r. You see? So that's why you have this eyeball. That's why you have all of them like this, you see? If you look at that and this, it's the same. Capital Y, capital R, and with the small y, small r. It's the same thing here. Capital R, Y, capital R, and with the small y, small r. So it's the same. But remember, we select them. Even this one is going to be the same. So that now when I combine this and this, you form this. This and this, you form this. This and this, you form capital Y, small r. And this and this and this, you see? This and the R, this and you see? Yes. Chico, are you getting it?
Can you please explain again?
That's why I'm going through slow here. So now, remember, when we're asking you in the paper, we don't ask you like this. We don't ask you like this. They will ask you, form the gametes here. Or they give the gametes and they ask you, what is the genotype? You understand? That's how we ask you an exam. And this is one of the part where students go off. But when it comes to exam, that's where student gets more marks because it is simple. So remember Punnett square? What they did here, the first part is right, is fine. So what they did is they crossed this and this. Then they produced this. But now because all of them are the same, I just picked one, put it here, I picked one, I put it here, then I crossed that and I produced all of them like this. Meaning that all 16 pairs, 16 individuals, all of them are going to be like that. Yes, George. All of them are going to be like that. So now what happens when okay, let me give you a best example. Someone's father and someone's mother, they get married. They produce someone's baby. They produce 16 babies. Those babies, they marry each other to produce this. That's why this is F1, P1, F1. First parent, first filial generation. So now when you get those kids you have produced, you marry them to again. It might not be possible in human beings, but it's very easy and possible in dogs and chickens and fish and what these other things is very possible. And they don't see it as awkward. However, in human beings, it is seen as awkward. All right. So no feelings here. So what happens here? They get those offsprings, they cross them together. That's why they cross this one. This crossed with, okay, let me just say, this offsprings crossed with the same offspring, capital Y, small y, capital R, small r, so that now they're able to get this. So look, When I bring that, each individual is going to form 4 gametes. First individual, remember the father is going to form 4 gametes and the mother is going to also form 4 gametes. So if I put them in a Punnett square, it means that our Punnett square is going to be like this. Let me draw it so that you're able to understand. There. So it means that now capital Y, capital R, capital Y, small y, capital R, and then small y, small r. So that is it. Yeah, I got them from here of the gametes I formed. And then now, so if this is the father, and then this is the mother. So it's gonna be capital R, capital Y, capital R, capital Y, capital, yes. Guys, I have to go down here so that I'm able to, yeah. Yes. Yeah. So if that's our Punnett square, so it means that it's gonna be, so the small y, capital R, and then small y, small r. So now, if that is our Punnett square, this is the Punnett square I'm talking about now. You see, I've put that in there. Now, what is next? It means that now I'm gonna, remember, I'm supposed to do this capital R, capital Y, like this goes with that, capital R, capital Y. And then I said this with that, this with that, this with that, and then I write. But because now this is a lot, it means that this is supposed, this one must go with the 4, this one must go with the 4, this one must go with the 4, this one must go with the 4. So it's gonna be too much and congested. So what we do, we use the Punnett square. So when we bring a question, they can fill in, they say X, Y, and Z. They ask you what is X, what is Y, and what is Z. That's it. Not too much stories when it comes to this. It's just different way how we teach it and different way how we set it. So this capital Y, capital Y, capital R, capital R, capital Y, capital R, small r. This with this, capital Y, small y, capital R. This with this, capital Y, small y, capital R, small r. Then this with this, capital Y, capital Y, capital Y, capital R, small r. So you see, you just have to go in filling it in. Once you fill it in, it's gonna come back like this. After that, you start counting. How did you get this 9 ratio? So it means that whenever you say capital Y, it is yellow. Whenever you say capital R, is round because they described it here. You understand? They described it there. So it means that this one is going to be yellow round. That's 1, 2, 3, 4, 5. This one, no. 6, 7, 8, 9. You see, it's 9. 9 is round and yellow. They are 9. Then we go to the next one. It's gonna be small y is green or black. Did they say green? They said black. Black and capital R is round. So it's gonna be 1, 2, 3. That's why you see a 3 here. So it's gonna be 3 black and round. So we are done with that. We go to the next one, which is capital Y, which is yellow, and small r. 2 of them, 2 small r's, which means is wrinkled, yellow and wrinkled. 1, 2, 3. So another 3 is gonna be yellow and wrinkled. You see, it's 3. It's 9 to 3, 3. Where is the 1? The 1 is here, which is black black and wrinkled, which is 1. That's why we got this ratio is 3, is 9 to 3 to 3 to 1. Guys, don't be disturbed by this too much explanation, because the way how we said it, you just need to know a simple concept. So when we go to the answering techniques of this question, that's when you're gonna open your mind much. So what you need to do is you just go back and then reread it. Do the reading. Do the reading. Yes. Then now I'm gonna send you some questions. Do those questions. Then now when we come back to the next class, then we shall see what we can do to see that we are able to pass this question. Yes. Examples of dihybrid cross. That is an example. You see, all this, we don't ask you all this, but just do that example. You do that example, you will see that something is building in your mind. Let me send it to you. Let me send it to you. You will do it. You will do it. You'll try to answer so that you're able to get all these answers. Yeah, so that's how we ask that question, you see. Let me first send you this. Yep. So it means that you have 3 questions you must answer. 3 questions you must answer. So guys, make sure that you answer those questions for me so that at least, yeah, it is easier for me to. Let me answer these questions and then we shall see what next. They're saying that certain plant species have the following alleles for each characteristic. For each characteristics, the number of seed per pod is capital P. And then one seed, capital P, and 3 seeds. It means that capital P, one seed is dominant over 3 seeds. Then the shape of the leaf, you see, number of seeds and then shape. So it's dihybrid. Normal, wrinkled. Then they're saying that the table below shows the results of offspring produced by a genetic cross between 2 plants of this species. Okay. How many genes of plant are considered here? Yeah, I need the answer from you guys. How many genes? Pensani.
There are 4 genes.
There are 4 genes.
I'm still looking.
Zintle is saying 4 genes. Pensani, what are you saying?
I'm saying I'm still looking.
You're still looking, ne? Okay. Georgie, how many genes are there? Georgie, I'm still waiting for your answer from the chat. I know you always send me answers from the chat. Hi guys, there are 2 genes, not 4. There are 4 alleles, 2 genes. Check. They say the shape. Remember, a gene has 2 alternative forms, either this or that. You understand? And then the number of seeds per pod is either this or that. It's either having 1 pod, 1 seed per pod, or 3 seeds per pod. The shape of the leaf, either normal or wrinkled. You understand? So basically there are 2 genes, 2 characteristics we are considering here. That's why it is a dihybrid cross. Is it clear? Then they're saying how many genes? So we are saying that is 2. Now name the dominant phenotype of the plant. Look, you look at the one which has a capital letter. In this case, we say that here, what is here? P, capital P is dominant over small p, meaning that one seed is dominant. Yes, so we get 1 mark there. And then when we come here, normal shape is dominant over wrinkled. So meaning that the next one is the normal shape. So those are the 2 marks which are supposed to be there. Do you see how it's easy to answer such question? Then they're saying that give the phenotype of each parent. They're saying give the genotype of each parent. So here, what are they looking for? They want the genotype of the parent. So here, what did they cross? So it's gonna be capital P. Yes, it's gonna be one seed normal, one seed wrinkled. Okay, 3 seeds. Okay, it's gonna be capital P, small p, capital L, small l. Yes, we get them from here. You understand? And then they're saying that name the offspring that are homozygous recessive for both alleles. Name the offspring that are, remember, offspring is 3 seeds, recessive is 3 seeds, and then wrinkled shape. 3 seeds, which is 3 seeds, wrinkled, 3 seeds, wrinkled shape. So it means that is this 3 seeds and then wrinkled leaves. So that is the genotype for homozygous recessive for both characteristics. Did I say? Yes. Yes. So yeah. Name the offspring that are homozygous recessive for both. Yes. So I think we got it. Then they're saying that, is there any other question? No. So I think we're gonna start with the genetic lineage here, or what you call the pedigree. Yeah, if we just do a small recap, not a recap, just a small hint. A box means a male, a circle means a female. So this means you married, and then this means you produce the babies. So all these are babies. So this one and this one are children of this, while this one just came into the family to get married, and then they produced these kids. The shaded one means is affected with that condition they have talked about. The unshaded one means that it is normal. So basically we will look at it when we meet again. So we need now to do questions concerning about this. Let me see if I sent, or that question I sent, everyone is, we have some questions there. Let me see if I have some questions there. We can practice in advance. Sandion, did you finish up all the questions I sent? Okay. Okay, I'm going to send it to you guys. You're gonna do for me the first. It's not that you're gonna do it, you're gonna understand it. It's just what is gonna be in the next topic, which we're gonna cover. So if you're reading ahead, you read ahead so that when we come, I explain and then we do those questions. So I'm gonna also send you questions for what we have discussed. So, but today you're gonna do for me those questions I sent now. Then tomorrow I'll send you the questions which we're gonna be covering in the next what? The next subtopic, starting from the pedigree. And then I'll look for more questions so that you're able to practice them and practice them. If you have a challenge, that's when you come back to me, and then I'm able to help you out if you have that challenge. Guys, if you have any question, Let me know, or otherwise I'm going to stop here. Yes, let me start with the Zintle. Do you have any question? You unmute, everyone unmute. But when I ask your question, you answer me. Okay, let me start with the Chico. Chico, do you have any question?
No questions.
Zintle, do you have any question?
Sir.
No question.
Yes, no question concerning this, but I do have a question.
You have a question? Is it a separate question?
No, I want to ask when are we starting what the ears, what are the ears, eyes, you know?
Now we starting with the ears, the what. Yeah.
Yeah.
So the next class we're gonna have is gonna finish the genetics. The next class. Just know that the next class we're gonna finish the whole topic of genetics. Then it means that we're gonna start the eye and ear. Those ones are not difficult, it's simple. Just one lesson we can cover up the whole structure of the eye plus the spinal cord. One lesson. Then the other lesson, then the eye will look at it as individual one lesson, and then the ear will also look at it as one as one individual lesson. So it means that you will have 4 lessons from now. Then it means that the ear will be covered. Then you will only remain with one big topic, that is the evolution. But evolution also have to be practiced. By the end of April, you'll be done with your syllabus. So it means that we'll just be doing revision from May. Is maximum revision. That's how I try to budget it, to see that by the beginning of May, the syllabus will be covered, everything covered. So it's only focusing on revision. Those who are writing in May, June, then you'll be able to um, write the paper. Before you write, still I'll see you in maximum time, maybe more than 2 or 3 hours. Then you go write. Then after that, paper 2, we also prepare for paper 2 again. We do drill. Then you go write. Then you come back with the distinction. Yes. Any other question? George said no question. Ario, any question?
No, sir.
All right. Okay, guys, have a lovely Sunday. See you again in the next class. If I find I have time, I'll just tell you that, guys, we might have a class. Yes, if I get a small time like this, I might even not even follow the timetable. I will just add more lessons to see that, yeah, you guys, you are covered, and I see that by the time you go to the papers, you are able to write and pass your exams. All right, guys, see you again the next
I'm
class.
having a class tomorrow.
For what? For life science?
Yes.
No, we might not have a class.
Okay.
Sharp.